So the electron transport chain happens a bit differently in prokaryotes. For one thing, prokaryotes don't have organelles, so they don't have mitochondria. The electron transport chain enzyme complexes and ATP synthase are embedded in the outer membrane. And so this is a very bad picture, right? This is my PowerPoint diagram, but this orange line is meant to be the plasma membrane, the cell membrane, phospholipid bilayer. This black is supposed to be the cell wall, that peptidoglycan cell wall, okay? And I don't mean outer membrane. So these are embedded just in the cell membrane. I didn't mean outer membrane. So the electron transport chain, enzyme complexes, and ATP synthase are embedded in the cell membrane. The protons are pumped by the enzyme complexes from inside the cytosol to outside the bacteria. And so you get these little red dots are meant to be the hydrogen ions that have been pumped from the center of the cell out to just outside the cell membrane. The ATP synthase allows these protons back into the bacterial cell and couples this with the generation of ATP. So this isn't going to be quite as efficient as in mitochondria because this cell wall is not truly keeping the hydrogen ions in. So a lot of these hydrogen ions are just going to dissipate, and that's going to make a less effective gradient. Now, if there is an outer membrane, as is the case with gram-negative bacteria, those hydrogen ions would be trapped, just like in the mitochondria, but they're not trapped here with just a cell wall. Prokaryotes also use a variety of enzyme complexes in their electron transport chains, and they don't all use NADH or FADH2 as electron donors. This is because NADH and FADH2 are generated by the breakdown of sugar for energy, but many bacteria use other kinds of energy sources, including inorganic energy sources, which would result in different electron donors. Also, in the absence of oxygen, some bacteria can use different final electron acceptors so that the electron transport chain can continue generating a very large amount of ATP in anaerobic condition. And I should say it differently. I said in the absence of oxygen, some bacteria can use different final electron acceptors. They can't switch. You either have an anaerobic electron transport chain or an aerobic one. The electron transport chains are set up to use one final electron acceptor only, and it can never use anything else. So there's not an organism that can switch back and forth between aerobic and anaerobic respiration. Okay, it uses one or the other. So some bacteria are always living in anaerobic conditions. They always use a different final electron acceptor. Aerobic and anaerobic respiration use the same three steps. They use glycolysis, citric acid cycle, and electron transport chain. And they produce similar amounts of ATP. The only difference is that oxygen is not the final electron acceptor in anaerobic respiration. Aerobic respiration produces the most ATP per sugar molecule, but other types of respiration can be similar. Okay? So usually if it's an anaerobic electron transport chain, the final electron acceptor will either be nitrate, nitrite, nitric oxide, sulfate, some oxygen-containing salt. Okay. But mostly it's the same. So everything we've covered about electron transport chains, whether they're aerobic or anaerobic, so yes, they might have something different than NADH and FADH2, but they'll have something that acts as an electron donor. They might have slightly different enzyme complexes, but they still will pump protons, right? They might have a different final electron acceptor, but they still have one. So the things, all that stuff that we just learned about aerobic respiration, it all applies to anaerobic respiration with very minor differences. Okay. So the reason aerobic respiration, the reason aerobic respiration produces the most ATP of all the different kinds of respiration is because atmospheric oxygen is the best oxidizer. Okay? That means it is the best at taking electrons even when they're in a very low energy, stable state. So because all of these final electron acceptors are not as good oxidizers, they're only able to clear electrons from an anaerobic electron transport chain if those electrons retain a little bit more energy and remain a little bit less stable. to allow their pickup. This means that in anaerobic electron transport chains, the electrons cannot give as much energy towards ATP production as they can when oxygen is the final electron acceptor. So oxygen can remove electrons even when they've given up every last bit of energy to ATP production. Even when they're in super stable state where they're at in the fourth enzyme complex, oxygen can still remove them. These other final electron acceptors could not remove electrons from an aerobic electron transport chain because they would have lost too much energy. They need to retain some energy to be picked up by one of these. So that's why you can't switch back and forth between different electron transport chains, because the aerobic electron transport chain that uses oxygen, those enzyme complexes extract more energy from those electrons so that oxygen can remove them, because oxygen could still remove them. It's not set up to be for electrons to be removed by anything else. Okay. All right, now let's think about the early Earth's conditions. Oxygen would have existed in the atmosphere, oxygen would not have existed in the atmosphere before photosynthesis evolved. We don't find oxygen on planets without life, right? Because it's generally created by photosynthesis. It requires, it's not an easy molecule to make. It exists in very low abundance on other planets. So oxygen would not have existed in the atmosphere before photosynthesis evolved. So oxygen was not available as the final electron acceptor. In these anaerobic conditions, the earliest microbes used fermentation, which we'll get to next. Fermentation is very inefficient. It's a very poor way to extract energy from food molecules, but it works acceptably, right? And then anaerobic respiration to generate ATP. We know that fermentation was first because essentially all cells can do it, because it really only requires the glycolysis step. All cells can perform glycolysis. It also does not require any special organelles or complicated enzymes. Fermentation generates very little ATP and a lot of acid. So acids are the byproduct of fermentation. So if we had fermenters first, after some time these fermenting microbes would have acidified their environments, increasing the risk that protons would leak into and acidify the cells. So when I say acidify the environment, I mean fill it with protons. That's what an acid is, something with a lot of protons, a liquid with a lot of protons. So as the waste, these cells are fermenting, they're excreting a lot of acid waste, they become more and more abundant on the planet, they start acidifying their environment. And that means that there's a high risk of these protons leaking into cells, right, like this, and acidifying the inside of the cell. Well, that's not so conducive to life. So microbes that developed proton pumps to maintain their internal acidity, their internal pH, to be more neutral, they would have been favored by natural selection, right? Because they can control the pH inside them. It was these proton pumps that then became useful in the evolution of the electron transport chain. Once these proton pumps were coordinated for the production of ATP, microbes could generate a surplus of energy and life could really take off on Earth. All right, so let's just do an electron transport chain recap with anaerobic electron transport chains in mind. Electron transport chains are driven by high energy electrons from sugars or other molecules. So those high energy electrons came from high energy molecules. The energy of these electrons is used to generate a proton gradient across a membrane. So it can be across the cell membrane for prokaryotes or across the inner mitochondrial membrane for eukaryotes. Aerobic organisms require oxygen. Well, then you have chemiosmosis. So protons flow back through ATP synthase, driving ATP synthesis, right? So with oxidative phosphorylation, aerobic organisms require oxygen to be the terminal electron acceptor. So oxygen will clear those electrons to keep that proton gradient going and keep chemiosmosis going. But in anaerobic electron transport chains, it could be a different final electron acceptor. So oxygen or another final electron acceptor is necessary to clear the electron transport chains so that electrons can keep moving through slowly, keep making that proton gradient, keep allowing ATP production by the movement of protons down that concentration gradient that was created. The electron transport chain generates most of the energy from respiration, whether it's aerobic or anaerobic, it doesn't really matter. Electron transport chains, whether they're aerobic or anaerobic, they generate far, far, far more ATP than glycolysis and the citric acid cycle combined.